Okay, so good afternoon, everyone. A couple of housekeeping things. So we've kind of more or less officially closed the book on term quiz 1. The marks came back. We had the rebuttals. I marked the rebuttals. So we're closing the book on term quiz 1 and getting prepared for term quiz 2, which takes place next Monday and Tuesday. So it'll focus on predominantly on the content from the last 2 weeks. So that was the kind of last part of the immune system, and then all of the nervous physiology stuff that we covered in the 3 lectures last week. So that'll be the focus, but you can expect that there might be a question or two that try to get you to connect that content from the last couple of weeks to some of the stuff we talked earlier about in the course. So you can't completely ignore everything you learned before, because again, the human body is very interconnected, and so you need to be able to see that interconnectivity and be prepared to draw those connections between different parts of the course. Other than that, the short communication assignments, I know I've started having conversations with people about ideas related to the questions they want to pursue. So that's great. I know that I have seen a few submissions to the AI peer reviewer, so hopefully it's actually being some kind of useful tool for you as well. And I also made sure that I let the TAs know that there are submissions starting to roll in. Some people have submitted their module 2, so I wanted to draw the TA's attention to that and ultimately get them aware that submissions are coming and they should be on the lookout, and hopefully you'll get some timely feedback from them on your assignment submission. Okay, so those are the only things I wanted to talk about. Are there any, excuse me, are there any questions about things before we continue on here with the lecture this afternoon? Everybody's all good? All right. So we ended the lecture on Friday talking a little bit about the microbiota gut-brain axis. And where we had gotten to really was this idea that if the intestinal epithelium becomes leaky, becomes permeable in some way, and we outlined a number of potential causes to the leakiness of that intestinal epithelium. If the intestinal epithelium becomes leaky in some way, then suddenly the commensal microbes that are in the gut lumen, and which would normally, if the intestinal epithelium were intact, be prevented from interacting with the immune cells of the body. If there is this leakiness that occurs, then ultimately these commensal bacteria, these commensal microbes, are able to enter into the body, and they now interact with the immune cells. And that causes the immune cells to begin producing cytokines, right? Again, we learned about that in the immune system lectures. They'll begin to pump out cytokines, and those cytokines can make their way through the circulatory system, ultimately to the brain, where they can exert effects on the brain. And so that's where we started talking about right at the very end of the lecture on Friday. So we have this figure here where it shows some of these peripherally released inflammatory cytokines. So these would be the cytokines that are being produced in the body by these immune cells when they have detected these commensal microbes that have come into the body across that leaky epithelium. And again, just trying to connect the dots between things we're learning now and things we've learned before. TNF alpha, interleukin 1, interleukin 6, the 3 cytokines they highlight there are the 3 cytokines that we talked about being produced by M1 macrophages, right, several weeks back. So we get these immune cells detecting these commensal microbes, pumping out these pro-inflammatory cytokines, and then when they make their way to the brain, they trigger the production of the same cytokines within the brain, right? And so now we see astrocytes and microglia within the brain producing their own pro-inflammatory cytokines in response to the peripheral pro-inflammatory cytokines that have come to the brain. And this is where the problems can come in. This is where this leaky gut can lead to changes in the way our brain functions, because this figure that I brought up now talks about neuroinflammation, right? So an inflammatory response within the nervous system. And you can see that on the right-hand side here, it talks about the negative aspects of neuroinflammation, right? And if we just kind of highlight some of the things here, anxiety and depression, cognitive impairment, reduced neuronal plasticity, neuronal damage. Yeah, these all sound like kind of negative things. And it mentions here that when you have either high or you have chronic levels of neuroinflammation, that's when you're going to get these negative aspects of the neuroinflammation response. So we can imagine that if you have this leaky gut, and especially if it's not just a transient condition, but something that becomes maybe more of a chronic long-term effect, then you're going to constantly have these commensal microbes from the gut, leaking in to the body through that compromised intestinal epithelium, interacting with the immune cells in the periphery of the body, triggering these pro-inflammatory cytokines to be produced that then trigger pro-inflammatory cytokine production in the brain, and that ultimately leads to all these negative things, right? Anxiety, depression, cognitive impairment, etc. Right? So if this becomes kind of a long-term chronic state for the body, then we're going to see these negative aspects. Now the figure does also highlight for us that there are positive aspects of neuroinflammation, right? And so there you can see things like enhanced plasticity and tissue repair and neuroprotection, right? All good things. But now you'll notice the words transient and low kind of predominate here, right? That it's when you have, short-term, low amounts of neuroinflammation, that's beneficial to the brain function. But when you have the chronic and high levels of neuroinflammation, that's when the negative effects predominate. And so this is part of what makes this a bit of a tricky thing, because there have been researchers who've contemplated, well, if we know that someone is experiencing this leaky gut and they're experiencing this chronic neuroinflammation within their brain, and this is impacting, again, their mood and their cognition and so forth, could we not try and treat this through some kind of anti-inflammatory? A corticosterone, NSAIDs, the things that we talked about earlier in the course. But the challenge there is that we don't want to knock the inflammation levels down so low as to not derive those positive neuroinflammatory benefits, right? So this is not a simple solution of saying, oh, we've got a leaky gut syndrome or a leaky gut situation, let's go use some anti-inflammatories to solve the problem, because that could be just as problematic and just as damaging to the brain if we knock down the inflammation to the point where we can't benefit from these positive aspects of neuroinflammation either, right? And so this is what makes the whole situation kind of a tricky phenomenon. So, perhaps going back to the list from earlier of the various causes of leaky gut, which can then lead to this alteration of brain function, really, it's another example of prevention being the best medicine here, right? That we need to try and figure out how can we, again, limit the stress that we are experiencing in our lives? How can we, alter our diets in ways that keep the fat levels down? Can we avoid things like excess alcohol consumption? Obviously, some of these things we can't avoid. I mean, you can't avoid getting pregnant necessarily. If you want to be pregnant, you're going to be pregnant. This is going to be a consequence, potentially. Strenuous exercise, right? I mean, again, we can limit our exercise, make sure that we're not going too hard at it. This is probably a case, again, of prevention being better than trying to use some kind of pharmacological treatment. Okay, continuing on with this figure here, again, this whole microbiota gut-brain axis, I want to focus now on the left-hand side of this figure, where it talks about the HPA axis. So this stands for the hypothalamus pituitary adrenal axis. This is basically one of the body's stress systems. the other being the sympathetic nervous system that we've talked about before. And what you'll notice is that the hypothalamus pituitary adrenal axis culminates in the secretion of cortisol. And there's a lot of evidence that when we are stressed and when our adrenal glands are pumping out cortisol, this stress hormone, this can actually alter the composition of the microbes in our gut in ways that could then feed back to lead to alterations in brain function again. And so one study I'll point out here, I chose this one because it seems kind of relevant to you. This is a study where researchers were measuring the abundance of bacteria within the gut of undergraduate students, kind of in relation to writing an exam. So you've got here on the y-axis colony-forming units, so just a standard measure of bacterial kind of abundance. And you can see here they measured the CFU on some baseline day, so some day far, far before any exams were being talked about. They then remeasured it on the day before the exam, on the day of the exam, and 5 days after the exam. And you notice the significant decline that took place beginning on the exam day and really on the days after. And so the conclusion these researchers came to is that, well, the examination process, as we all know, is a stressful process. So your cortisol levels spike on the exam day. And in some way or another, these cortisol levels going up because of the stress is actually negatively impacting the commensal microbes that are in your gut. It's actually causing the commensal microbes to begin dying. Right? And that's why the CFU value is dropping here significantly and remains down for several days even after the exam is over. Right? Even after you might not be so stressed out now, you're still experiencing these lingering effects of there being a decline in the commensal microbe levels in the gut. And so why exactly does this happen? Well, the prevailing hypothesis is that cortisol impacts the secretion of the gut. So cortisol is thought to, for example, reduce the secretion of acid by the gut. It's thought to decrease the digestive enzymes that are secreted by the gut. It's even, as they kind of point out here, maybe more indirectly, cortisol shuts down the immune system. And so the kinds of immune signaling molecules that are being secreted into the gut may also change in response to stress. And the thinking is that as the gut environment changes now because of this decrease in acid and enzyme and immune signaling secretion, it may create an environment that is no longer hospitable for the commensal microbes. And so they begin to die. Right? Commensal microbes presumably are going to thrive under the normal conditions that prevail within the gut, right? Under acidic conditions with digestive enzymes there and lots of immune signaling compounds. As you have this stress response turn on and you start to have cortisol altering gut secretion and immune cell system secretion, you're changing the environment within the gut lumen in ways that may not be hospitable to the commensal microbes, and so they begin to perish, and you see their levels decline. This can become problematic in part because, even thinking back to our talk about commensal microbes from earlier, one of the things commensal microbes do is they help to keep out pathogenic microbes. Right? That they occupy the real estate and kind of make sure there's no territory that the pathogens can take over. And so if the stress response creates a condition that is not hospitable to the commensal microbes and their levels decline, this could create a scenario where pathogenic microbes, if they are present in the environment, could now colonize the gut. And this could be part of the reason why so many people will note how they get kind of sick in the days following a stress, right? You have a stressor, you were perfectly fine maybe for a couple days afterwards, and now as you're starting to relax, that's when you're starting to experience the illness. It could be that the stress knocked down those commensal microbes, and then you get a few days for the pathogens to get in there, and start to proliferate and take up the space the commensals once occupied, and then you begin to feel those illnesses several days after the stressful event has passed. Okay, the last part of this diagram I want to explore here is this kind of bottom or extreme right-hand side over here, where it's talking about the production of short-chain fatty acids by the commensal microbes, and these too can have profound impacts on brain function. I'm going to swap in a different figure here to talk about this, a little bit more detailed figure. So let's start off by what the hell are short-chain fatty acids? Well, as their name would suggest, they are literally just, short-chain fatty acids, right? They are 2 or 3 or 4 carbon long fatty acids. So you see them pictured and named here. We're talking about things like acetate and propionate and butyrate, right? These would be the most common of the short-chain fatty acids. So where do these short-chain fatty acids come from? How are they related to the gut microbes? The answer is they come ultimately from complex carbohydrates in our diet, what we often refer to as dietary fibers. So we're predominantly talking about compounds like cellulose, right, that we get from plants. This would be a great example of a complex carbohydrate or a dietary fiber. And the thing about cellulose is that our human body does not produce the enzymes necessary to digest it, right? We as humans do not produce a cellulase enzyme to digest cellulose. And so if not for the action of the gut microbes on these cellulose that we are eating, that cellulose would just pass through. And indeed, even in the presence of gut microbes that can digest that cellulose, we still get a lot of it passing through, which is actually vitally important because that's part of what creates the bulk of our feces, right? And sometimes when people are not getting enough dietary fiber, they start to experience things like constipation, right? Because ultimately there's just not enough undigested material to kind of collect up into feces and defecate. So dietary fiber, super important even for just regularity of bowel movements. But what's happening here is these complex carbohydrates like cellulose, we again cannot digest them, but the gut microbes, they do make cellulase. And so they are able to chop the cellulose into its individual glucose, and then they actually pick up that glucose, they absorb it into their body, the microbes do, and they put it through fermentation. They put it through glycolysis, followed by some fermentation process at the end, so they can derive energy from it. Right? So they benefit from these dietary fibers. This is how we feed our gut microbiome. But then the end products of those glycolysis fermentation reactions the microbes do are these short-chain fatty acids. The microbes can't oxidize these any further because you have to have access to oxygen to actually metabolize these short-chain fatty acids further. And the gut lumen is a very anaerobic environment, so it's mostly colonized by various kinds of anaerobic microbes. So they have gotten as much energy from the glucose as they could. They have produced these short-chain fatty acids as a waste product of that process, and they just dump them back into the gut lumen. Now at that point, a couple of things can happen. One is we can absorb these short-chain fatty acids into our body. And so here they show what's called the monocarboxylate transporter, MCT. So it's going to allow for these short-chain fatty acids, which, as you can see, have a single carboxylic acid group in their structure. It allows for them to be absorbed into the body. And then from there, they can be circulated throughout the body. And that means they can get to the brain, which we're going to talk more about in a second. The other thing is that our gut epithelial cells have just receptors on their cell surface, and they kind of name some of the receptors here. And when the short-chain fatty acids bind to those receptors, it can trigger the gut epithelial cells to release various kinds of signaling molecules into circulation, and then these signaling molecules can exert effects on the brain. So either the short-chain fatty acids are being absorbed and they're going directly to the brain and exerting effects, or they're basically triggering the release of signaling molecules within the gut, and those signaling molecules transit to the brain and exert effects. Either way, whether direct or indirect, the figure on the right-hand side here shows us all the benefits of these short-chain fatty acids on our brain. And if you look through the list here, it's like increased neurogenesis, so being able to make new neurons, improved cognitive development, improved memory, reduced inflammatory signaling, improved integrity of the blood-brain barrier. These are all good things. These are all things that are going to improve our cognitive health. So the long and short of this story is that we need commensal microbes in our gut. We need dietary fibers in our diet so we can get these short-chain fatty acids that ultimately are going to promote good brain health, okay? And so again, we have to have dietary fiber in our diet to benefit from this. The recommended daily intake for fiber is 25 grams a day, right? And so this is one of the things that's on the nutrition labels when you're buying food, right? I challenge you for the next little while when you're eating things to have a look at the nutrition facts and assess, am I getting today the 25 grams of dietary fiber that I need? Because if I'm not, you know, one of the things that I might be suffering as a result is I might not be getting these brain benefits, right? Everybody wants to do super well in this course. Everybody wants to do super well in all their courses. You know, boosting your dietary fiber to boost your short-chain fatty acid production could certainly help with that. I also want to just show you a little bit of empirical data here just to kind of demonstrate that, again, I'm not just like talking out my asshole here. There's some data that we can show here. Right, this is a mouse study. Yeah, you're hoping I don't talk about my asshole because she's behind me most of the lecture here, right? So anyways, yeah, there's a little bit of data here from the mouse study I can show you. So when you want to test the memory capacity of mice, one of the most common ways this is done is this experimental design here. You basically put a mouse into a room and you place two identical objects into that room, and you give the mice about 10 minutes to kind of wander about and explore those objects, right? They'll go over and check one out, they'll go over and check the other out, they'll kind of keep checking these things out. So you give them 10 minutes to explore these objects, and then you take them out of the cage, and you wait for an hour, and then you put them back in, right? And now when you put them back, you have two objects again. One is a familiar object they saw before, and one is a novel object that they've not seen before. If they remember the familiar object, if they have good memory, they'll spend most of their time exploring the new object, and they'll just ignore the familiar one, right? And so we can very simply test the memory of mice by doing this kind of experiment and measuring how much time they spend with the novel object relative to the familiar one. Right? If they spend way more time with the novel object than the familiar object in that second part, it's because they remember that familiar object from before. There's no need to explore it again. They've already seen it. They know all about it. So here we have a study where they were measuring what they call here the discrimination index. So just a measure of how much time did they spend with the new object compared to the familiar object. And you can see here that if they were given short-chain fatty acids, so if they were injected with short-chain fatty acids, that actually doubled their discrimination index compared to the control, right? So they were way better at remembering things if they had been administered these short-chain fatty acids. This is the proof of things I was saying before, that short-chain fatty acids can boost the memory function of the brain. So that's some empirical data. You know, one of the things that's been hypothesized then related to this is we know that there is cognitive decline with age. You know, you just have to talk to the elderly people in your lives, and you know they're not quite as sharp as they once were. And you know, one hypothesis is that this may be due to declines in short-chain fatty acid production that may occur with age. Right? It's possible that maybe the gut environment becomes less hospitable over time to microbes. The microbes can't crank out those short-chain fatty acids anymore. You're not getting that boost effect on the brain. This is one hypothesis that certainly is being explored. The other thing I want to point out here, because it was measured in this mouse study, is where it talks about EE. This is environmental enrichment. And so you can see that it was just as beneficial as short-chain fatty acids, giving the mice an environment that's enriched to live in on a day-to-day basis. This is what this would look like, right? If you imagine, I mean, probably not quite this elaborate in a research setting, but basically if you've ever had pet rodents, you know, hamsters, mice, rats, whatever, you know that you have to put little toys and things in the cage, give them something to do. They found in this study that just giving environmental enrichment did help to boost the memory function of these mice. So it's, you know, you can sort of mimic, shall we say, the effects of short-chain fatty acids through environmental enrichment. And again, this is likely the same thing in the human case. You know, one of the places where I think this becomes most relevant is when you think about individuals living in long-term care, right? You know, I don't know if you've ever been to a long-term care home. It can sometimes be a pretty boring place. I know that they try their best to try and provide all kinds of activities. You know, today we're going to play this game or that game or coloring time or whatever. And that engagement is super critical because without it, you know, cognitive decline is likely to hasten, right? One of the pieces of advice I can give you here, especially as your parents get older, is make sure that you know, talk to them routinely, right? Provide them with something to do in their days. You know, my parents are getting up there. My dad's almost 80. My mom's going to turn 70 next year. You know, sometimes they just tend, I mean, at least they're very social, but ultimately, I try to call them, you know, days when I'm traveling back and forth down the highway just to talk to them, just to give some enrichment to their day. Yeah, they do lots of social things, great. But yeah, if that stops, if you start to see that okay, they're not as social as they once were, they're not going out and doing as many things, you know, when you start to see that happening in a person, it's not usually too long after that that cognitive decline starts to really set in. Okay, so let's switch gears a little bit here now. We're going to move out of the organization of the nervous system and move into the other part of this lecture, which is the cells of the nervous system. This light micrograph image I have here shows the two major cell types in the nervous system. The first one, the one that's very obvious here, occupying most of the space, is the neuron. Right? So neurons, of course, are the cells of the nervous system that are directly responsible for its functions. So when you think back to our very first slide and all the various functions of the nervous system, sensory input, motor output, integration, memory, and all the associated capacities that endows the human body, it is the neurons that are directly responsible for those functions. I added onto this slide a few important properties we should know about neurons. One is that they are amitotic, which means they do not undergo cell division, right? And so this is the basis of your parents warning you, you know, like, don't do that, it's going to kill brain cells and they don't come back. You know, if you've ever been threatened in that way, it's true, right? Brain cells are amitotic. In most brain regions, when a neuron dies, you know, it's not like the neighboring neuron can just undergo mitosis and be like, oh, here, I got you. Here's a new neuron. Now, you might then say to yourself, well, earlier you were telling me about short-chain fatty acids and how they increase neurogenesis. Where do the new neurons come from in neurogenesis if not from original neurons? Well, they come from stem cell populations in discrete regions of the brain. Right? So there are stem cells in a few locations in the brain, mostly locations associated with memory and learning, and new neurons are produced by those stem cells. So it's not through the division of existing neurons. Corresponding to their amitotic nature, neurons are typically long-lived, so they have a very long lifespan. They have to because, again, they're generally not mitotic and can't be replaced easily. And then lastly, they have incredibly high oxygen and glucose demands, right? It's often said that, you know, your brain maybe accounts for, what, 2 or 3% of your body weight, yet it accounts for about 20% of your metabolic rate, right? Your brain is a heavy hitter when it comes to energy demand. It is consuming tons of oxygen and glucose, right? And the reasons for that, I think, will become clearer as we transition into talking about electrophysiology towards the end of the week. The other cell type we find within the nervous system are the so-called neuroglia. So all those little small purple cells you're seeing there are neuroglia. They're probably just the nucleus that you're seeing there. Neuroglia are kind of interesting for a couple of reasons. One, the name neuroglia translates from Latin to mean nerve glue. And it reflects the fact that when people first discovered these cell types in the nervous system, they thought their function was entirely structural. They thought that their job was just to be the glue that holds the nervous system together, so they called them neuroglia. As we're going to learn a little bit later in the week, this is not true. Yes, there is a structural role for some of these neuroglia, but there are multiple types of neuroglia, and they actually play a lot of important physiological roles, kind of helping to assist the neurons with their function. So we've come a long way in terms of our understanding of these cells. A couple of properties about them. So unlike the neurons, the neuroglia are mitotic. They can undergo cell division to create more of themselves. They are far smaller, as you can already tell just from the image, but they're also far more numerous. And so in fact, as my comment here in the top now says, within our brains, there are about 5 to 50-fold more neuroglia than there are neurons. Right? They're way more numerous. However, because the microglia are so much smaller than the neurons, even though there's up to 50-fold more microglia in our brain, the microglia occupy about half of our brain's volume and the neurons the other half. So we have many fewer neurons, but because they're larger, they occupy the same total volume as the smaller, more numerous neuroglia. I want to move on now and talk a little bit about the structure of neurons. And so I start with this little figure here. This comes from kind of an old textbook, but it's a beautiful image showing us the structure of neurons within the mammalian brain. And I put this up here just so you can see, again, the structural diversity that exists within neurons or among neurons, right? So just even looking at this image, you can appreciate that there are so many different, structures that neurons can have, and this again is just in one location within, the nervous system, the brain. So yeah, just so you can kind of marvel at the beautiful diversity here. But of course, as good scientists, we get a little bit frightened by diversity. We want to try and classify things into groups to make it easier for us to comprehend. And so I want to introduce you then next to the structural classification scheme that we typically use to talk about the structure of neurons. So the way to kind of introduce you to this structural classification scheme, the way that it's basically structured is it's based around the idea of how many processes are sticking off the neuronal cell body. So the cell body is the location of the cell that houses the nucleus. So in this neuron here, there's the cell body right there. You can tell because that's where the nucleus is. We classify neurons on the basis of how many processes are sticking off that cell body. And so we're going to start over here on the right-hand side of this figure where they're talking about multipolar neurons. So multipolar in this case means that there are 3 or more processes sticking off the cell body. Well, let's look. There's our cell body where the nucleus is. I count 1, 2, 3, 4, 5, 6 processes sticking off that cell body. That's definitely more than 3 or more. That makes it a multipolar neuron. Now, the figure also goes on to show us that there are 2 major locations in our nervous system where we find multipolar neurons. One is that efferent neurons are typically multipolar. You might remember that the efferent division of the nervous system is the motor division. So we're saying motor neurons are typically multipolar. Why is that the case? Well, the major, benefit, shall we say, major characteristic of multipolar neurons is because they have so many dendrites, they have so many of these processes sticking off of them, they're able to receive multiple sensory inputs simultaneously, right? And so you typically find multipolar neurons present in parts of the nervous system where you need a high level of integration, right? Where you have a lot of sensory input that needs to come into a cell and be integrated into a single output. So if we think about, as an example, our motor neurons, so let's imagine this is a motor neuron that is stimulating muscles within your arm, right? Maybe muscles that you can use to like extend your arm to punch somebody, right? You can think to yourself, okay, well, there's probably a lot of factors that are going to go into determining whether I want to punch somebody in the face right now, right? And so I need all these different pieces of sensory information from other brain regions to come together, so I decide, am I going to stimulate this muscle and punch her or not? I won't. I do like her. I'm not going to punch her, all right? Anyways, but that's the idea, right? You have all these different pieces of sensory information, right? I could be thinking about, I'm trying to think of all the factors that could lead me to want to punch or not punch her, but, you get the idea, I hope, that there's all this sensory input that we need to be able to integrate together to make a decision about kind of, do I actually punch this arm out or not, right? So multipolar neurons are great for times where you have a lot of sensory input that needs to be integrated to make a decision about, a particular output or not. So in the case of muscles, again, we can think about how we want to not just have our motor cortex necessarily just send signals and say, hey, let's punch, right? We want to be able to have input from higher brain structures to say, ethically it's not right for me to punch her, or, whatever, maybe I'm afraid of something, I'm afraid I'll lose my job, or whatever the case might be. There are other inputs aside from just my brain saying, extend your arm, you could punch her, that need to be regulated and integrated here. The same is true because, of course, they highlight here that multipolar neurons are found also in the central nervous system. And this, of course, would never surprise you because if multipolar neurons are all about integrating sensory information, I mean, this is what the whole damn central nervous system is really about, right? We're sending all kinds of sensory information from all over the body to the central nervous system to be integrated, right? Think about last week when we were talking about those polymodal association cortices in our brain, right, that are receiving all kinds of different sensory input and trying to put it all together, again, to kind of help us have a holistic picture of what is happening in our environment and within our body. We need neurons in the central nervous system to be able to receive all kinds of different sensory input, integrate it all together, and then make a decision about whether or what kind of output we should generate. Now, also in the central nervous system, they talk here about what are called anaxonic neurons. So anaxonic literally means like no axon. And if you look at this drawing of an anaxonic neuron here, you kind of see what they are meaning, right? That when you look at those several processes that are sticking off that neuron cell body, it's not evident that one of them is different than the others, right? Typically, when we think about neurons, you think, okay, there's one axon, right? A neuron has one axon, and in this multipolar case, it was pretty clear which was the axon. Here again, pretty clear what the axon is. But in an anaxonic neuron, it's not clear that there's an axon. It looks like they're just a bunch of dendrites. As people have started to do more kind of molecular and biochemical characterization of these particular neurons, they've come to realize that actually an anaxonic neuron, the name has now become a bit of a misnomer because the thinking is every one of these processes is both a dendrite and an axon. And so what's kind of cool about the anaxonic neurons is instead of having kind of a directionality of information flow, right, where we typically think of information comes in at the dendrites, flows out through the axons, it's kind of a one-way system, the thinking with these anaxonic neurons is information can kind of flow in and out in all kinds of different ways, right? And therefore, we can use these neurons kind of like old-fashioned telephone operators of sorts, right? If you imagine an old-fashioned telephone operator, receiving calls and routing them out to somewhere, then receiving calls from that place and routing it back to where it came from, this ability to just kind of receive and send signals in all kinds of different directions, then these are pretty cool kinds of neurons, right? They're not just sending information in a unidirectional way from point A to point B, but they have this capacity to relay information perhaps bidirectionally or multi-directionally, depending on how many processes there are. Lastly, we come to what are called the bipolar and then the pseudo-unipolar neurons, which you see predominate within the sensory division of the nervous system. Bipolar, so again, if we look at the cell body, there are two processes that are sticking off the cell body. So that's where bipolar comes from. And on the pseudo-unipolar side, obviously unipolar refers to one process. So we see that here. Here's the cell body. Just one process sticks off of it. Why we call these pseudo-unipolar is because developmentally, these actually begin as bipolar neurons, and then over development, the cell body kind of moves to the side so that the two processes kind of blend together. And so we use the term pseudo-unipolar to say, hey, these are not true unipolar neurons. A true unipolar neuron would only ever have one process. These guys had two processes when they started developing. The cell body then kind of, shifts to the side. The processes merge together, so it looks like there's just one, hence the term pseudo, like false unipolar. Now, the key characteristic of both the pseudo-unipolar and the bipolar neurons is that, the cell body, these are not very highly integrating neurons. I guess that's the point I'll make, right? Especially that's evident in the pseudo-unipolar ones because, here's your sensory input coming in through the dendrites. I mean, the dendrites lead directly to an axon. The axon bypasses the cell body. There's no integration of sensory information happening here. So this is the exact opposite of the multipolar, where it's like integration is key. These pseudo-unipolar ones are the opposite. They're like, we don't want to do any kind of integration of sensory information. Their job is to collect sensory information either directly or through being linked to some kind of sensory receptor, and then they are going to just rapidly relay that information to somewhere else in the nervous system. It's not about integration with these pseudo-unipolar and bipolar neurons. It's really about detect the sensation, rapidly relay that sensory information to other places within the nervous system. All right, so even in the bipolar case where you do have your cell body still kind of in the way here and the signal has to pass through, there's going to be probably minimal integration of that signal really happening. More than you'd get with a pseudo-unipolar for sure, but not as much as you get with a true multipolar neuron. The other thing I want to highlight about these sensory neurons in comparison to the multipolar type is you'll notice how the size of the dendritic trees here are typically very small in sensory neurons, right? And especially if you contrast that, I mean, look at the dendritic tree of this multipolar neuron here, which is massive, and even this one here, it's quite an extensive kind of dendritic surface area. With sensory neurons, your dendritic surface area, the size of that dendritic tree, is very limited. Right? And that's to allow for precision in sensation, right? It's what allows us to be able to identify precisely where a piece of sensory information is coming from. Think about the importance of this in vision, for example, right? When you have all these photons of light from your environment coming to your eyes, I need to be able to know precisely where every individual photon is coming from for me to have a clear picture of what's happening in this room. If I have big dendritic trees in my sensory neurons, like my visual neurons, then I'm going to end up with more of a kind of blurry vision, right? A very poor resolution vision where I can see stuff, I can see colors and things out there, but they would probably blend together. Like if I take my glasses off and you guys just become a blur of colors and faceless entities out there, right? We need to be able to have just narrow dendritic trees so that every sensory neuron, we can sort of say, okay, Well, if we've got a whole bunch of these bipolar neurons all lined up side by side by side by side by side, we can tell that if light struck here, right, we can distinguish that from light striking here because it's two separate neurons picking up that sensation, right? We'll spend just a little bit of time here, I guess, with the last few minutes we have, just kind of introducing you to this slide here. I really like this slide as a way of thinking about the structure of neurons because it kind of simplifies things down in a very kind of functional, physiological way. This figure basically highlights for us that a typical neuron has three major parts to it. The first part is what they call here the dendritic zone. And its function is to receive sensory input. And what you'll notice, the dendritic zone here is kind of colored in yellow in this figure. What you'll notice is there are two kind of major parts to the dendritic zone. One is the cell body, or the soma as it's known, and the other is the dendrites. And what I really like about this figure and this way of thinking about neurons is it really reminds us that the cell body is the kind of sensory input region of the neuron, and that the dendrites are merely just extensions of the cell body, right? The whole purpose of dendrites is just to increase the sensory receptive area of a neuron. I think sometimes we put too much emphasis on the cell body and the dendrite as being somehow separate structures, but from a functional point of view especially, the dendrites are just extensions of the cell body of the neuron. And so the cell body itself, is receptive of sensory input. You can see in this picture, these green things here are meant to be, not sperm. They kind of look like they could be sperm, like confused sperm or something, but they're meant to be the axon terminals of other neurons. You can see that they often will interface directly with the cell body, right? They don't always interface with just the dendrites. So the cell body itself is receiving sensory input. The dendrites just work to extend the total surface area of the cell body so you can increase the total receptive area of the neuron. Now, of course, if you've got a ton of sensory information coming into a given neuron's dendritic zone here, it needs to be integrated together, right? And so they highlight here for us a region of the dendritic zone called the axon hillock, right? It's the part of the dendritic zone right at the interface with the axon. And this is where all the sensory information that is being received by this neuron's dendritic zone, it's all sent to the axon hillock, and that's where the integration takes place. We're going to talk in more detail later in the course about how exactly that happens, but this is the location of integration of sensory input within the dendritic zone. Next we have the axon, and it says here the axon conducts excitation. So in a nervous system parlance, the word excitation is code for action potentials. So axons conduct action potentials, but axons also generate action potentials. You should probably add that word on there. Generates and conducts the action potentials. And where the action potentials are first generated is this first region of the axon right here called the axon initial segment. And so what happens here is the integrated sensory input from the axon hillock is basically fed forward to the axon initial segment, and then it decides whether there is sufficient sensory input to generate an action potential or not. And if it generates an action potential, then the axon conducts that action potential faithfully along its length and brings it ultimately to the final part here called the telodendritic zone. And its job is to send out information, right? Very commonly it does this in the form of neurotransmitters, right? And so what we see here in the telodendritic zone is how the axon branches. So we get what are called the axonal terminal branches here. This allows for this one neuron to then output signals to a whole bunch of either other neurons or muscle cells or glandular cells. And what's interesting then too is that, the fact that you have these dendrites on one end, right, expanding the surface area of the dendritic zone, and then you have these axon terminal branches on the other end, allowing for signals to be output to various other neurons or muscles or glands. As it points out here, neurons, a single neuron could be interacting with as many as 60,000 other neurons or other structures, right? So the branching that we see on the dendrites and the axon terminals allow for an extensive amount of integration to occur. Okay, we'll stop there. We'll continue on Thursday, go into a little bit more detail on the neuronal structure here. I'll see you then.